I'll be talking mostly about collaborations that extend far beyond and across disciplines. Nevertheless, it's nice to point out the Zakim Bridge that's part of the Big Dig that's now a decade old, but we have adopted for some years as a metaphor for the translational work of neurobiology to therapeutics for Parkinson's disease. In highlighting these translational themes and starting not in a non-standard way at least for pharmaceutical development, I'll review the evolutionary biology that's been the initial source for targeting urate, and then move to the epidemiology that's advanced it along with, very briefly, some of the neurobiological underpinnings for this target, and then update you on where we are in developing urate elevating strategy as a therapeutic for the disease. Just very briefly, at a biochemical level, urate is a purine, and in the context of purine metabolism, for example, adenosine in mammals is deaminated to inosine, to which we'll return, which then loses the sugar group to become hypoxanthine and then gets successively oxidized by xanthine oxidase to become urate, and that's where the metabolism of purines ends in humans and most primates, but actually in all other mammals, urate is metabolized by urate oxidase to allantoin. That step is lost during primate evolution. It looks like in an important way because of multiple mutations that occurred across the lineages of lesser apes like gibbons and gorillas and chimpanzees, amongst the greater apes and us, leading to higher levels of urate some 10 to 15 million years ago, it seems, for reasons that are still not well understood, but have raised a number of interesting hypotheses. I won't go into much detail, that they haven't been sorted out. The cognitive benefit of higher urate is something that was born out of the similarity, as I mentioned, urate is a purine, just like caffeine. There's actually very interesting work that suggests there may indeed be cognitive benefits to higher levels of urate, even if unsubstantiated. Medically, there's a notion that's been developed that higher levels of urate may have provided an advantage in raising blood pressure, although, as it turns out from some of our and others' recent data, an hypertensive effect may not be borne out, but nevertheless, it's raised the possibility that there may be medical reasons for which, or systemic blood pressure reasons for which higher urate levels may have increased during our evolution. Probably most compelling, but still not yet proven, almost a half a century ago, it was proposed that because we lost the ability to make our own endogenous antioxidant vitamin C, at around the same time we lost the ability to break down urate, that urate might serve as an antioxidant function, which was demonstrated quite convincingly about 40 years ago by Bruce Ames and colleagues, showing that urate is a potent antioxidant and comparable to that of vitamin C, except that for the reasons I mentioned, we have much higher levels of urate. And that's where really the prospects for targeting urate in Parkinson's was taken up by epidemiologists. And I'll go through some of the data here, but first I'll introduce you across town in Boston to my colleague, born in Milan actually, Alberto Escario, and his epidemiology group with whom we've collaborated over the years, in this case over urate, and his work substantiating that of others, that in a very well-designed prospective cohorts, in this case the health professional follow-up study, in which in this some 50,000 men were followed for a number of decades, and those who provided blood samples were examined years later for the development of Parkinson's disease. And what was found was that if one just takes at baseline in this prospective epidemiological cohort, folks who had lower but normal up to the uppermost quartile of urate values and look to see who developed Parkinson's disease. Compared to those in the lowest quartile, there's a clear and very significant trend, especially above the median of urate values, for a lower risk of developing Parkinson's disease. In the study that was done and a number of other studies as well where this has been borne out, one can look at blood levels at least 4 years before diagnosis and to address the possibility that having early Parkinson's alters urate levels. And to the contrary, the evidence has become even more robust, as you can see here, looking at increasing urate levels and the risk of developing Parkinson's disease. This is just a relatively early meta-analysis. There have been a number of other prospective studies that have shown that if you look at the relative risk of developing Parkinson's disease over time as a function of baseline urate levels, those who have higher levels of urate are less likely have a reduced risk of developing Parkinson's disease. This, on the dietary end, just extracts from a cohort, not their urate levels, which weren't available in all the health professional follow-up study cohort members, but just their diets, and looking at diets that are so-called uricogenic, that increase the likelihood of raising urate levels or having higher urate levels. A uricogenic diet split into quintiles. Again, you can see this relationship. At diets that generate higher levels of urate, there's a decreasing risk for developing Parkinson's disease. And that led us to ask the question if amongst healthy individuals a higher level of urate predicts a reduced risk of developing the disease, how about people who already have Parkinson's disease, although they have, on average, a lower level of blood urate, there's still a wide range, and one can ask whether higher levels of urate in early Parkinson's disease predict a slower rate of progression. And to do that, we took advantage of collaboration with our colleagues in the Parkinson's Study Group of North America, convened now some 30 years ago, in pursuing clinical trials for Parkinson's disease, particularly for disease-modifying therapies, with as yet no clear successes. Nevertheless, the group has collected a lot of data and pursued the philosophy of trying to learn as much as we can about the disease by sharing these data. And what we were able to do was to go back to a couple of studies. The first one we approached was the PRECEPT study, which tested an anti-apoptotic drug, cephalon 1347, that turned out to be unsuccessful in slowing the rate of disease. But in that study, some 800 subjects were followed for a couple of years very meticulously early in the disease at a stage when they were not yet requiring dopaminergic therapy. Again, although randomization to the anti-apoptotic drug didn't slow down the disease, we had baseline data and serial data on rates of progression as well as safety labs. And one of the labs that was assessed at baseline in the PRECEPT study was urate. And we were able to stratify, again at baseline. Here are quintiles, as you can see, increasing. Just to the right, you can see what the normal in milligram per deciliter levels are, just a typical range of urate levels. And looking here at the whole group, you can see that the likelihood of, this is the hazard ratio for reaching the primary endpoint, which in the PRECEPT study was time to disability warranting dopaminergic therapy. And the likelihood after 2 years of going on dopaminergic therapy was half that in people with higher but still normal levels of urate compared to those in the lowest quintile. And the trend across the group was quite significant. In men, it was even more robust and quite significant. In women, there was a trend, but because women tend to have lower levels of urate, substantially lower levels, it's not clear that the lack of significance in women was related to the fact that there are so few women in the upper ranges of normal urate for men and women combined. Or whether there's a biological difference, something that comes up as we've moved through clinical development. But I'll just point out also in the PRECEPT study, which had as a potential surrogate for clinical progression, imaging biomarker with dopamine transporter ligand uptake, here with beta-CIT scan, beta-CIT uptake. This slide just illustrating that it can potentially be used as a marker of progression within an individual in whom over years and in correlation with worsening, increasing UPDRS scores, one can observe a decrease in the uptake of dopamine transporter binding ligand in the striatum. And these are those PRECEPT data plotted again with increasing but normal levels of urate, plotted against the rate of losing dopamine transporter ligand uptake over the 2 years of the study. And one can see that particularly at the highest quintile, about 7 to 8 milligrams per deciliter, those who entered with that higher level of urate lost their dopamine transporter binding sites as assessed by beta-CI uptake at a significantly lower rate. Again, was robust in men, and there was a trend, but much more noise in women. So with this initial look, we saw that a higher serum urate could predict a slower rate of clinical progression, as well as a slower risk of the disease developing in the first place, and raised the possibility that urate or one of its determinants could be an approach for disease modification. But before we got there, we had the chance to actually go back to that original photo, which of the PRECEPT investigators back in the mid-1980s that had emblazoned across their shirts the very first large-scale disease modification study really done, but that was done by the PSG as well, the DataTop study, where we had initially hoped to ask this same question, but didn't think urate was available in blood samples until it was found on a floppy disk back in someone's office, and we're able to assess whether higher urate levels may also be predictive of slower progression in this cohort. Similarly, about 800 subjects were followed for a couple of years for progression. In that case, it was selegiline and vitamin E that were tested and found not to be effective, but in a cohort that was very rigorously followed. In addition, in the DataTop study, a serial CSF was collected, and we were able to measure back in the lab CSF urate levels, and I'm not showing the data here, but very much like the blood urate levels, higher data, and these have been published a few years ago now, higher CSF urate levels were also predictive of a slower rate of progression. These are the serum urate levels combining the values from both the DataTop and PRECEPT studies for the rate of progression that was the primary outcome for both studies, again, as a function of at enrollment serum urate levels. And you can see this relationship where over the normal range of serum urate, it's fairly flat until about the median of the population, and then there's a significant and substantial reduced likelihood of progression early in Parkinson's disease with higher but normal levels of urate. So these data showed both from epidemiological and clinical cohorts that urate could be a predictor of favorable outcomes. Without belaboring it, but just to highlight over the last 10 years, the robustness with which urate as a biomarker has been linked to favorable outcomes in the disease. I started with pointing out some of the epidemiology early in the spectrum of disease progression with a number of studies showing higher levels, less likely to get the disease. In terms of progression as well, even there was some recent data with REM behavior disorder showing that people with higher levels of urate are even more likely to quickly go on to, higher levels were less likely to go on, lower levels more quickly to go on to Parkinson's disease from REM behavior disorder. I just showed you some of the prognostic studies. There was a very nice complementary study to the PRECEPT and DataTop cohorts, which I mentioned out of Sorrento and I believe Precious Mochia and colleagues who found that higher levels of urate in a cohort followed longitudinally was also a predictor of favorable outcomes in terms of non-motor symptoms of Parkinson's disease. And then I mentioned that the dietary influences on urate are also a predictor of higher urate on a lower risk of developing disease. There are also genetic data for major determinants of serum urate. Serum urate in humans is a mix determined by both genetic and environmental factors. Most recently, Alberto's group also found that genetic determinants of higher urate were also predictors of a slower rate of progression in a so-called Mendelian randomization study. All converging to highlight that urate could be a predictor of favorable outcomes in the disease. For want of time, primarily, entirely, I won't go into detail about the substantial biological data that have shown, to some extent, not surprisingly, that urate is a major antioxidant, is capable in disease models, animal models, cell culture models of conferring protection, models of Parkinson's disease. But I'll just highlight that there's now a mounting body of data that show that biologically it's plausible that urate may not be just a predictor of favorable outcomes, but could be a mediator of those favorable outcomes to the extent it's neuroprotective. This is actually work, I think the first in Parkinson's primary dopaminergic neuron cell culture model from Patrick Michel and colleagues in Paris, that just show in a dose-response curve, this is just cell culture dopaminergic neurons, their survival is dependent on urate in the culture, such that across a range of concentrations that are typical for concentrations in the brain of humans, higher levels of urate are protective. And here, by way of highlighting some of the work that has continued in our lab and in other labs, I'm just going to introduce some of the folks who've conducted some of the work. This is Sarah Cipriani from Firenze, who did some of our earliest work in cell culture. These are just to summarize her findings, both in primary dopaminergic cell lines, as well as, dopaminergic neurons in culture, as well as cell lines, finding that urate could be protective, and interestingly finding, somewhat surprisingly, that urate's effects through astrocytes are required to protect dopaminergic neurons in cell culture models. This is Rachit Bakshi, who subsequently went on to not only confirm the astrocyte dependence using like Sarah had done with conditioned medium experiments, showing that if you treat astrocytes, that urate has a very modest effect of protection on dopaminergic neurons and dopaminergic cells on its own, but in the presence of astrocytes, it's quite robust. And if one treats astrocytes with urate, they elaborate something in conditioned medium that's quite protective of dopaminergic neurons. Rachit found that it turns out it's through activation of a pathway called the NRF2, NRF2 pathway, and release of glutathione that urate confers protection in these cell culture models. And then Shichuan Chen took an in vivo approach, sort of recapitulating human evolution in mice in using urate oxidase knockout animals. We are urate oxidase knockout animals, which have much higher levels of urate, or conversely, urate oxidase transgenic mice that overexpress it with respectively high levels and low levels of urate, not only in the blood but in brain, and found that in those settings, in vivo models of Parkinson's disease, increasing urate levels was protective and decreasing them exacerbating. So these data show biologically that urate is plausibly a protectant as well as a marker for favorable outcomes, both based on its antioxidant capacity and other mechanisms still being explored preclinically. But really that was enough with the unusual convergence of these clinical, epidemiological, and laboratory data to suggest that targeting higher urate elevation would be worth exploring clinically. At the outset, we had to decide how to try to do that. Urate itself, it turns out, doesn't get across the gut-blood barrier. And we didn't have the option of blocking its breakdown because evolutionarily we've lost those enzymes that break it down. There's an interesting possibility of altering transport, which is actually a major determinant of by altering clearance of urate to raise levels. But we ended up initially taking the most straightforward approach, which was just a metabolic precursor approach using inosine, which, as I mentioned, is the deamination product of adenosine and upstream of urate, and initiating clinical studies with that precursor. And so in the last minutes, I'd like to go through how far that's come in the last few years with the completion of a phase 2 study and a phase 3 disease modification design study now beginning. The phase 2 study, which focused on safety of urate elevation of Parkinson's disease, or SHORE-PD for short. As most know medically who are familiar with urate, it's a rather small, little-known secret that urate is the main antioxidant that circulates in humans, but it's much better known for the side effect of having lost that enzyme during evolution and high urate levels, which is our risk for urate coming out of solution and forming crystals, and if that happens in a joint, it can be extremely painful, as in gout classically, and also kidney stones. And we wanted to keep that in mind as when we were focusing initially on elevating urate in Parkinson's patients. But our long view was actually very much informed by the unusual availability of human data to help us design how to elevate urate. And basically, what we really and are hoping to do is to recapitulate those early studies that linked higher levels of urate to better outcomes here in progression of Parkinson's disease. So the general design to which we're working is one in which we start with people who have faster progression of the disease on average, with lower levels of urate, enrolling them and then randomizing them to either placebo, in which case they'd stay below the median, or at least in phase 2, we compared dosing, not specific amounts of inosine, the precursor, but inosine dose with frequent blood level checks, titration to serum urate, to basically bring people up to 6 to 7 or 7 to 8 milligrams per deciliter. And that's what we did in our phase 2 study. On the safety end, it was very clear that for major adverse effects, people in whom we used this strategy to elevate urate did not develop major side effects. In fact, had fewer than those who were in placebo. Nevertheless, with the crystallopathic adverse events to which we were sensitized, we paid close attention, and while in gout, there were no cases of episodes that developed. We did get dose-dependently, although in a few people. This was a small study of 75 subjects, followed for 2 years with placebo or inosine to elevate urate. But in those in the mild to moderate elevation groups, there were 1 or 2 kidney stones, at least one of which was a uric acid kidney stone. We've learned a lot, and we're hopeful that with this strategy, including, for example, identifying stone formers early, uric acid stone formers early, by tracking their crystals in their urine sediment to keep the likelihood of such adverse effects low if we're able to go forward. Tolerability was excellent for inosine in that no one had to reduce their doses. This is just sort of proof of concept using inosine, not urate, that we could actually elevate urate levels in blood and CSF. Here's that range that's typical of from 4 to 8 milligrams per deciliter. We included only people who are below about 6 milligrams per deciliter, and then these are their averages at screening for several visits, and then they were followed for up to 2 years before a washout. Placebo group, of course, stayed fairly low, in their urate levels, but we very effectively were able to elevate into the 6 to 7 range or the 7 to 8 range urate levels by dosing and titrating to inosine. This was a correlate, as it turns out, antioxidant capacity, which you can measure in a number of ways here by an iron-based antioxidant capacity, or FRAP assay, and we were able to see that just like the urate elevation, we, in the mild to moderate urate elevation groups, saw a highly significant elevation in the antioxidant capacity in blood. In CSF, this shows that significantly inosine was able to elevate CSF levels. It was only done on a single visit and in a subset, but still quite significant. Lastly, from that phase 2 SHORE-PD study, although the main outcomes were safety, tolerability, and urate elevation, which were clearly met, we were reassured to see that there was at least a trend in a not adequate, with no intention of powering to look at progression. we saw at least data that were consistent with UPDRS changing over time, the possibility that it could be lower in people who are on the moderate dose. This has all led us to enthusiastically propose moving to a much larger disease modification design study that we're hoping to begin in just a few months, again taking advantage of the Parkinson's Study Group, which is distributed across North America. This is just to let you know where we are with this. This is work that's, the phase 2 was supported by the Michael J. Fox Foundation with a substantial investment, as is being made by the National Institute of Neurological Disease and Stroke at NIH in the US, with some additional support from the Fox Foundation. We're making drug, actually, as it turns out, in Milan. Inosine is being manufactured, and then it'll be assembled into the placebo and clinical trials material in the US. And in parallel, we're doing some FDA mandated clinical pharmacology studies, which will need to be done before we start, but we hope to start enrolling what will be around the springtime of next year, and hopefully over the coming years we'll be able to see if this non-traditional approach might be helpful for disease modification in Parkinson's disease. And I just wanted to also acknowledge those who are funding the work as well, and thank you for your attention.